Thank you for joining us today. I'm going to be talking to you about how we advance multimodal research on our Cancer Genomics Cloud platform, and a pretty great use case for this, for using deep learning applications on pan-cancer PDX, pediatric-derived xenograft histology. First, I want to do a short introduction on who we are as a company. Velsera, created recently, is really the merging of three different companies that's really expanded our offerings and expertise in the biotech field, as well as what we can offer, such as today, what we're offering in machine learning. So the agenda for today is we're going to go over the goals very briefly, how the PDX community or the patient-derived xenograft community is using machine learning, the resources and tools that we have on the Cancer Genomics Cloud that lend itself towards using machine learning for these outcomes, and then to go over what are the goals for machine learning and diagnosis, prognosis, and treatment in oncology before our conclusion. So from this webinar, I really want you to understand how the community is using multimodal research, including machine learning, what the resources and tools required to do this research are on our Cancer Genomics Cloud platform, as well as understand what the clinically centric goals are for machine learning tools in oncology. So how are we using machine learning? Well, first we need to define some of the tools and definitions that we have in the field. So in traditional machine learning methods, to not get caught up in, I don't want to get caught up in the figure, but really to just take away that there are about four main classes of machine learning methodology. There are the classifiers, which are usually yes or no answers, or feature extraction by separating features by the widest margin. Then you have the more complex deep learning, where each neural node is doing something very specific, and then each neuron within the network is coming together for an outcome, such as a prediction for how well a patient may do with treatment, or prediction on whether, given an X-ray or a histological sample, is this cancer, or is this a tumor or not? And so it uses a lot of imaging data for these. Time series data is much the same, and except that it uses a series of images, and it can also use genomic sequences, so that it uses genomic data as well as imaging data together in order to make a more informed decision. And then the neural networks, the graph neural networks, which also work similarly, but what they are utilized for many times is cell morphology or nuclei morphology. So it'll take one cell on a slide or nuclei on a slide and compare it to its neighbor, and then that neighbor to its neighbor, creating edges, and then making predictions based off of the morphology of the cells or the cell makeup of the histological sample. So how do we apply these machine learning methods to clinical outcomes? Well, one of the ways that we can do this, or that has been done, is using machine learning methods on oncology clinical research. And so a popular model for oncology research are patient-derived xenografts. And what these are is you take a few cancer or tumor cells from the patient, you implant them into the niche or into the mouse, and then you treat that mouse with interventions or drugs and see how it responds, either in survival time or tumor growth, or the proliferation or reduction in certain cell markers that may indicate cancer. And what Xi et al. Showed in 2017 is that when they took these histological slides and used them as training data sets for a machine learning algorithm, that it was able to predict the tumor type pretty well, but that it was going to require a lot of data. And this is where the Cancer Imaging Archive and the Imaging Data Commons within the PDX network came in. So what we did is we did a open call for everyone who was a part of the PDXNet consortia, all of the research centers. So Baylor College of Medicine and Jackson Labs, for instance, all submitted their histological images to us, to Velsera, and we put them on the platform. And then with the consortium and collaboration, we did a lot of standardization and cleaning of the data to make sure it's good data, right? Because for any machine learning model that you have a training data set for, right? It's really garbage in, garbage out. So you really have to make sure you have a strong set of data going in. And so we did a lot of work on that end. And then we made the images publicly available on the Cancer Genomics Cloud, and also created some tools that not only allowed us to check the data, but also serve to create publication-ready images for the researchers. And this had some great outcomes. One of the publications showed that using HoverNet, one of the tools on the CGC, that HoverNet can pretty closely to a human pathologist estimate the cell type and the cancer epithelial, which are cancerous cells and which are not, and what the cell types are, pretty close to what a human pathologist can do. And this is great news because pathologists are often backlogged and overworked. And so any kind of tool that can help them catch up or take some of that load off is really going to be a great and strong tool. And it also showed, and this is just kind of the methodology for how HoverNet sees the images. It does regional classification, and it looks at the morphology of the tissue and segments those out, and then makes a decision on whether which regions are necrotic, which regions are stromal cells, such as vasculature or support cells, and which cells are tumor cells. One other tool to come out of this was a deep learning model that could, from mouse tissue, accurately tell which tissue samples had, or which xenograft transplants had gone through lymphoproliferative disorder. And so what that is it's a rare condition. You see it in patients, but you also, see it in mice with these transplants. And it's when it involves immunosuppression following an organ transplant. And what it can do is it can mask the original phenotype and function of the tumor types that were originally implanted. So, the results you're getting may not or are not accurate. And so having a deep learning model that was developed that can do quality control, having a tool that can do quality control for your slides to also help check the data was something to come out of here, which was really great. If I had something like this in graduate school, I probably could have shaved off a year or so and saved myself gray hair. And so what are some of the imaging tools that we have that are related to PDXs? PDX imaging tools do we have available on the Cancer Genomics Cloud? So as previously mentioned, one of the strengths of the Cancer Genomics Cloud and using these tools and data is that we have a large repository of ready-to-use images. We have whole exome and RNA-seq data, and we have the tools and apps that are publicly available, but we usually add some type of ease of use to it for using it on our platform, such as a graphical user interface, for example, because a lot of these are code-based. So for one of the first tools to mention is HistoQC. It does in a automated and quantifiable way quality control to identify any artifacts on your histological slides. And what this does is handling the data and the slides upstream and doing that quality control for you upstream, then makes it better for downstream application and other machine learning tools. So it's really adding to the research and NIH goals of reproducibility and robustness of data. And it identifies a lot of outliers and it has a very interactable user interface. And if you prefer to use just the code base itself, with all of these tools, you can also use just the code base if that's what you're more comfortable with. And so the next tool is the star of the talk, which is HoverNet. And what HoverNet does is it does that simultaneous image segmentation in that cell or nuclei neighbor comparison in order to tell you which cell types are there and make those predictions based off of histological images. And so the purpose of this is to separate the clustered nuclei that you often find on the histological images in order to better predict what cell types are there for sampling. And so, for example, and it depends on the tissue type. So, for example, if you had a breast cancer tissue sample, then it would classify tumor, lymphocyte, or stromal cells in the tissue. And then this last tool was actually started out as a module on GoogleNet. It's called Inception V3. And it provides deep learning for, again, cell segmentation. And so doing kind of a similar neural network application as the previous tool, it gives out artifacts or can tell you what type of cell is in the tissue through a different type of neural network. And so we do have more tools than just histology. Machine learning is a large field within the life sciences that is gaining traction. And so there's been a lot of other applications that we've been able to add to our platform. And just to remind you that the Cancer Genomics Cloud has petabytes of data in which to use these tools with, and you can always upload your own data, which will not be made publicly available unless you choose to, but you can use your own data as well with some of these other data sets to compare. And so with there's a lot of multi-omics tools, genomics, proteomics, metabolomics, along with the imaging, machine learning tools. And so you have about 800 different tools and public projects with which to do your analyses. And one of the really cool machine learning tools is for single cell measurements. And what's called MC-MICO. And so what it is it's an end-to-end whole slide processing tool in collaboration with the NCI, who are always developing real best practices. It's supposed to act as an end-to-end single-cell RNA-seq and temporal expression by area of the cell platform. So you can do everything within one tool on the CGC platform. And then one of the other really great tools that we have is the image viewer. So what the image viewer does is it uses DICOM images. Those are digital imaging and communication. So that's like X-rays, MRIs, those types of things that you would get done at a hospital or from a doctor when something is suspected, or if it wants to get or if you need a diagnosis. And so the image viewer allows you to interactively explore these images and see what the predictions are. And there's new data sets being added to it. It actually has a very large testing data set already. And you can analyze images with a GUI or with an API, as I previously mentioned. And the NCI is also closely collaborating with that open source server of the DICOM images to keep the standard tools for analyzing the images up to date. And so what are some of the advances in machine learning and oncology that have been done by the community? Well, first, we have to understand what the goals of deep learning for oncology are in supporting risk diagnosis and prognosis when selecting treatment for the patient. And the workflow for this usually is that you have a patient population, you get an image from them, either a histological image or one of those DICOM images. It does the segmentation and the tiling to do those comparisons within the tissue, fed into the machine learning model, and then the model makes a prediction. And so the first step of using machine learning in oncology, in an oncological clinical setting, is risk stratification to understand what the patient's risk is for developing cancer. And one of the examples used here is breast density. And so what it uses is a 4-dimensional where it adds time as well as structure to a breast scan. And it's fed into a machine learning model in order to detect, based off of breast density and time of and time of the scan, is the patient at risk for developing breast cancer. And this may be combined with also the patient's, genomic information. And then the next step and goal is diagnosis. So it's making an accurate diagnosis of the sample. So this is another machine module that the previous may be fed into. It's meant to catch errors, help time-constrained physicians, and there's constantly improved accuracy and consistent evaluation, right? So you don't want the outcomes changing every time you feed the same image in or feed a test data set in. And the goals for the methods are for it to be a non-invasive way, so that someone doesn't always need a biopsy, for example, that may be painful or take a lot of recovery time. Instead, if they can have images taken and used by a machine learning model or tool, then they can have a more accurate or just as accurate diagnosis. And so some of the strengths of these tools have been a large abundance of training data. Images are all over the place and available, and they're usually multimodal models. So they incorporate a lot of other analyses and tools into them. So it's not just one object making the decisions or doing the analysis. And some of the future directions that are being done right now are their evaluation in a clinical setting, so a live evaluation, as well as a prospective evaluation where pathologists are using the program or not using the program, and then comparing the results and speed with which they can make an accurate diagnosis. And lastly is prognosis and treatment selection. So this is selecting the correct, most effective treatment for the patient, hopefully without bombarding them with different drugs that can be damaging, but not as helpful, and trying to find an effective treatment. And so you want to find that a patient's best survival outcome as well as best treatment options. And there's a variety of models that work together in this, and getting a patient their treatment selection right. There's radiotherapy planning. There's thinking about what the response to treatment is going to be, what their prognosis for survival is, what are their genomics, what do the images look like, what are the tumor features. And all of these are working together when figuring out a patient's treatment plan. And usually by people. If you have a machine learning tool, in this example, this one is ResNet 50, it can make a lot of those predictions for you, taking the load off of the treatment team and getting you those treatments that are effective much faster. And so conclusions, first, there are applications of machine learning across biology. Big fields, obviously, are genomics, as well as virology, especially when you have something like COVID, which shares symptoms with a lot of other viral infections. Then having a tool that can quickly take a patient sample and say, this is a cold, this is COVID, this is the flu, you can imagine is very helpful for the next pandemic. And as well as applying these machine learning tools for predictions of metabolic diseases, so diabetes, or what the consequences can be for a patient given their current metabolic status. And so what I want you to walk away from this webinar with is that you can think of the Cancer Genomics Cloud as a multimodal research hub where you can do multi-omics, genomics, proteomics, and machine learning-based image analysis. And that collaboratively, lessons that we have learned on the way here is that it's going to require a lot of collaboration with scientists. That's data gathering, data analysis, working together so that we can verify results. And a lot of these have already resulted in, a lot of collaborative communities and community use. So the scientists are really on board with sharing and having like centralized areas for shared commonly used tools and data for analysis. Also, the NCI is heavily invested in imaging support, and that's why we work so closely with them on the CGC. A lot of the data that they make available is also available on the CGC for you to readily use. And that lastly, supporting translational science is the primary goal of the CGC, and what we hope for these machine learning tools is to get these from the basic research or the lab research side to actually being able to apply them in the clinic and get patients where they need to be much better, much faster than is currently being done. So I want to acknowledge our team, Dennis Dean, our team lead, yours truly. Background is in genomic sciences. Saul Acevedo, who's our data scientist, and his background is in modeling statistics, and our previous team member who really did a lot to get us going when we first started taking up a lot of these machine learning challenges, which was Sonar, and as well as the multi-omics team campaign, the CGC team, research adoption education, product development, as well as Sarah, Rowan, and Zelia for helping to organize and put this webinar together. And thank you.